Hey everyone, thanks for joining me today for episode 869 of People Behind the Science. I'm your host, Dr. Marie McNeely, and today I am joined by our guest, Dr. Ashok Shetty, to talk about life and science. This episode is made possible with support from our sponsor, Innovative Research. Listeners, Innovative Research has provided scientists with quality lab products made in Novi, Michigan for more than 20 years. Their portfolio includes human and animal biologics, assay and ELISA kits, antibodies, and more. Products can be purchased directly from their website by credit card or purchase order at www.innov-research.com. That's www.innov-research.com. They offer small quantities, bulk purchases, and custom orders. From human whole blood to specialized antibodies, Innovative Research has what you need to succeed in the lab. And today, our guest Ash is going to talk more about his lab and his life. So listeners, get ready to meet another one of our outstanding people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am thrilled to be speaking with our guest scientist, Dr. Ashok Shetty. So, Ash, welcome to the show today. How are you?
I'm doing great. Thank you for having me.
Well, Ash, thank you so much for joining us, and we're looking forward to learning more about you and your work. But first, let me take a moment to tell our listeners more about you and how you got to where you are today. So, listeners, Ash is a university distinguished professor in the Department of Cell Biology and Genetics, an associate director of the Institute for Regenerative Medicine at Texas A&M University, Naresh Vashisht College of Medicine. Ash earned his Ph.D. in neuroscience from the All India Institute of Medical Sciences in New Delhi, and he completed postdoctoral research at Montana State University and Duke University. Afterwards, Ash joined the faculty at Duke University in the Division of Neurosurgery, and he joined the faculty at Texas A&M University College of Medicine in 2011. In 2024, Ash was honored with the University Distinguished Professor Award from Texas A&M University, and he's also received the College of Medicine's Senior Research Excellence Award. In addition, Ash is a fellow of the American Association for the Advancement of Science and the American Society for Neural Transplantation and Repair. He has received the Research Career Scientist Award from the United States Department of Veterans Affairs. He's been recognized among the world's top 1% of scientists across all scientific fields, and he was the 2025 honoree of Fast Company's World-Changing Ideas. And in our interview today, Ash, we're excited to get to know you as a scientist, of course, but we also want to know more about your life outside the lab and more about you as a person. So can you share, what do you like to do when you're not doing science?
So I tend to work a lot and spend most of my time on my job. Managing my research and doing science keeps me very busy. Being a scientist is a job that never really stops. And most of us scientists don't really complain because we chose this path and knew what it would be like. But when I do get some free time, I enjoy being with my family and getting some exercise. I also like playing brain games like Sudoku, cut tiles, or solving puzzles. Sometimes I go out to see a popular movie at the theater. Otherwise, it's mostly my job.
Mostly work. So what do you like to do for exercise when you're able to get some time to yourself?
So I do mostly cycling, stationary cycle, and I do it for about 30 minutes. So I can do 6 to 7 miles in that 30 minutes.
Well, it's great to get a window into your life outside the lab. And I think you're absolutely right that science can sometimes be all-consuming and you feel like you're working quite a bit.
Yes. It's a 24/7 job.
Oh, exactly. I think sometimes it doesn't feel painful, though, because you are so excited about the work that you do and you're enthusiastic to go in every day.
Exactly. So when you love your job, it's not really work. You just enjoy it.
Definitely. So let's talk a little bit more about your science. Ash, how do you describe what you do to someone who is outside of your specific field or perhaps just outside of science altogether?
Explaining what I do in science can be tricky, especially to people who are not scientists. So many of scientists, they are not used to talking about our research in a lay language. But over time, I have learned how to share my work in a way that's easy for anyone to understand. I work on developing treatment for neurological and neurodegenerative disorders using stem cells and stem cell derived products. We call it extracellular vesicles. These are tiny vesicles secreted by stem cells, but they carry the secretome of stem cells. So we call it the goodies. These goodies are scientifically termed as microRNAs and proteins, but once we administer them into the brain, so they can induce significant beneficial changes in various neural cell types in the brain. So that can improve brain function.
Well, Ash, I'm excited to get into some of the details about your research as we go through our conversation today, but I think it's really important to talk about motivation as well. As we've alluded to, there's a lot of work to do in science, and it can be quite a busy career, but I think being excited about it and being motivated to do the work that you do can be really empowering. So do you have a favorite quote or a saying or something that just motivates you every day?
Yeah, I like motivational quotes. I read them often. My favorite quotes are one from Thomas Edison. Opportunity is missed by most people because it is dressed in overalls and looks like work.
I like that one.
Yeah. And I also like the another one from Roosevelt. This is very relevant to what we are doing in science. Believe you can and you are halfway there. I always remember that when I'm venturing into new projects.
Definitely. I think that optimism can go a long way.
Yes. And another quote from Steve Jobs is also my favorite. The people who are crazy enough to think they can change the world are actually the ones who do.
I think that is a fantastic quote as well. I love these 3 motivational quotes. Well, I think having these forces that motivate and inspire you is really important in science, but also having the right people around you can make a tremendous difference. So for you, when you look back over the course of your career, were there particular people, whether they were role models or mentors or just these inspirational figures that you looked up to, who had a big impact on you in your career?
I have had several role models at different points in my career. The first is my Ph.D. supervisor, Dr. Gomati Gopinath. She was at the All India Institute of Medical Sciences, New Delhi, India. And she was a disciplinarian, but also a wonderful teacher. She actually taught me research skills from the ground up. And more importantly, she demonstrated me how focus, hard work, perseverance, and resilience are key in succeeding in research. So in the beginning of my career, I learned quite a lot of things from her. And then another scientist I really admire is Dr. Fred Rusty Gage, who was the president of the Salk Institute until recently. I have never worked with him or collaborated with him, but he has been one of my favorite scientists since my PhD days. So I followed his research throughout my entire career. So my research topics such as neural plasticity, cell therapy, hippocampal neurogenesis, and brain aging lined up closely with what his lab has been doing for many decades. I always looked forward to reading the original research articles coming from his lab, and then I presented them at journal clubs. I was always very impressed with the quality, rigor, and creativity of their work. He is still one of my favorite neuroscientists.
Oh, I love that. And I think having both these mentors, like you described in your PhD advisor, who kind of show you the ropes, but also these inspirational figures or role models who you model your career after and follow their work can be really helpful as you're developing your career, particularly in those early stages. And I'd love to talk a little bit more about what those early stages were like for you, Ash. So can you take us all the way back to the beginning and share with us, how did you first get interested in science to begin with?
Becoming a scientist was not something I planned. I grew up in a small rural town in southern India in a middle-class family. Back then, just getting a job after finishing a bachelor's degree was a big deal where I'm from. So my dad was a school teacher and he hoped I would land an even better job after college. So maybe that's why I thought of becoming a college lecturer, which sounded like a great next step. It felt like moving up from being a school teacher. So after my bachelor's, I went for a master's in medical sciences at a medical school, which luckily wasn't very far from my home. I majored in human anatomy, mostly because it pretty much guaranteed me a lecturer job at the same medical school after graduating. So I finished my master's at the top of my class. And as planned, I joined the medical school's faculty and started teaching human anatomy, including neuroanatomy to first-year medical students. So neuroanatomy quickly became my favorite subject. So once I finished what the textbooks had to offer, my curiosity led me to the library and where I started reading original research articles published in various journals. So I was fascinated by how incredibly complex the brain is, both in terms of its structure as well as function. So everyone used to say at that time, we know very little about the brain. And that just made me even more curious. So I realized that if I ever did research, neuroscience would be the field for me. But back then, there weren't many places in India offering a Ph.D. program in neuroscience. So the top one was the All India Institute of Medical Sciences in New Delhi, which is in North India. As I mentioned earlier, I'm from southern part of India, so it's a long way. But I still applied and got in after going through a written exam and an in-person interview. And I was lucky enough to join a well-funded and well-equipped lab doing cutting-edge research in cell therapy. So my thesis topic was on embryonic dopaminergic cell transplants, particularly studying their differentiation, migration, and integration in the adult brain. My PhD experience was very positive, actually, thanks to the support of my advisor, doctoral committee members, and fellow PhD students in the lab. So it was very, very positive. After PhD, I started looking for postdoc positions abroad. That's mainly because at the time, India just didn't have enough research labs or resources for new PhDs to keep doing research. I didn't want to go back to teaching. I really wanted to do research. So that's why I applied basically all over the world. I got postdoc offers from several countries, the United States, England, France, and Canada. But I decided to head to the United States. So my first stop was Montana State University in Bozeman, Montana, where I spent about a year and a half studying how prenatal alcohol exposure affects brain development. So things were going well in Montana, but I missed working on cell therapy. So at the Society for Neuroscience conference in 1991, I networked with scientists performing neural cell transplantation and ended up landing a postdoc position at Duke University Medical Center at North Carolina. And then moving to Duke was a huge shift. I suddenly found myself surrounded by big names in neuroscience. I worked with a clinician scientist. He's a neurosurgeon, Dr. Dennis Turner, who gave me the freedom to pursue pretty much any research I was interested in. So that really opened a lot of doors for me. I focused on brain repair research using neural cell transplants to study how the brain heals after injuries, especially focusing on the hippocampus. I also worked with both adult and aged brain models, and eventually this led me to research on status epilepticus as well as chronic epilepsy. So as my research picked up steam, I got the chance to join the faculty at Duke, starting as an assistant professor. And shortly after, I was awarded an R01 grant from the National Institute of Health and a merit grant from the Department of Veterans Affairs. And these were key in helping me set up my own independent research program. Then with more grants and a growing list of publications, I moved up the ladder to become an associate professor and then became a full professor in 2004. So when neural stem cells became a major focus in science, I jumped in and started working on postnatal neural stem cells, adult neurogenesis, and postnatal neural stem cell therapy for temporal lobe epilepsy and brain injury. So these topics became my main research areas for many years because I also had a part-time job at the Durham Veterans Affairs Medical Center. I also got the chance to work in military medicine, particularly studying the pathogenesis of Gulf War illness. So this eventually led me to test different drugs in this model. And then in 2011, I moved to Texas A&M University College of Medicine as a professor and director of neurosciences when they started the new institute called the Institute for Regenerative Medicine, headed by one of the well-known stem cell biologists, Dr. Darwin Prokop. He actually recruited me to Texas A&M University. My research on induced pluripotent stem cells and pluripotent stem cell-derived neural cells stem cell-derived extracellular vesicles and Gulf War illness, flourished at Texas A&M University. I was able to secure significant research funding from the NIH, Department of Defense, and Department of Veterans Affairs, which helped my work grow even further. So in 2024, I was honored to be named as a distinguished professor at Texas A&M University. So that's pretty much been my journey as a scientist so far.
Absolutely. And you make it sound like every step was sort of a logical progression, but I know that's often not the case.
Yes, I agree.
So when you think back, how did you make those decisions, or what kind of helped you figure out that next step of the career path as you were going through it yourself?
Basically, I took one step at a time, didn't really plan the entire tiers, but it just happened. I was successful in getting step one done. Then I went to step two. Like that, I moved up in my career.
I think it goes back to the quote that you mentioned, those opportunities disguised as work, right?
Exactly.
Well, it sounds like you have a fantastic lab and research program there at Texas A&M, and I'd love to talk a little bit more about your research there. So, Ash, is there a particular project that you're working on at the moment that you are just the most excited about and want to share with all of us?
One of the ongoing projects I'm excited about is testing the promise of human neural stem cell-derived extracellular vesicles in models of Alzheimer's disease. As you know, Alzheimer's is a devastating brain condition that affects about 7 million Americans and over 60 million people worldwide. So this disease has an enormous emotional and physical burden on both patients and their families. And unfortunately, there is no effective treatment for Alzheimer's. So our lab is developing an innovative biologic that could be administered nasally. So the biologic contains tiny particles called extracellular vesicles secreted by human neural stem cells. So these vesicles are naturally packed with powerful anti-inflammatory and neuroprotective molecules like microRNAs and proteins. These extracellular vesicles can calm down the overactive responses of the brain's immune cells. There are two types of cells which can cause inflammation in the brain. They're called microglia and astrocytes. So these cells, under physiological conditions, they help neurons. The neurons are the brain cells that transmit information through electrical signals. But in Alzheimer's disease, microglia and astrocytes become overactive and they trigger harmful inflammation in response to abnormal proteins that build up in the brain. So these problematic proteins include amyloid beta 42, and they form plaques, which are clumps of sticky protein that accumulate between neurons or brain cells. And you also see tangles, and these are twisted strands of another protein called tau that build up inside neurons. So by calming harmful inflammation, these neural stem cell-derived extracellular vesicles can protect the brain from further damage. So in a study published in 2024 in the Journal of Extracellular Vesicles, our research team tested these EVs in an animal model that mimics early onset Alzheimer's disease, which is the form of the disease that typically affects people under age 65. So we administered neural stem cell-derived extracellular vesicles intranasally during the early stages of the disease that resulted in a substantial reduction in brain inflammation. More importantly, the treatment didn't interfere with the microglia's normal function, which is phagocytosis. So it simply calmed the overactivation of microglia. So this therapy actually delayed the progression of cognitive decline. It preserved memory and thinking abilities for longer periods. It also helped prevent some of the mood disturbances such as depression and anxiety that commonly affect people in Alzheimer's. So in a follow-up study in 2025, we published another paper, and it showed that these stem cell-derived extracellular vesicles can protect human neurons from malfunctioning and dying when exposed to amyloid beta 42 oligomers, so which is the highly toxic form of the sticky protein that damages brain cells in Alzheimer's disease. And we published another paper this year, so using a model of brain aging, and which demonstrated that intranasal administration of neural stem cell-derived extracellular vesicles during late middle age can lessen cognitive decline or the so-called brain fog typically seen in old age, again by suppressing microglia-mediated inflammatory signaling pathways which are popularly called as neuroinflammation. So these findings overall suggest that intranasal administration of stem cell-derived vesicles in middle age could serve as a wellness strategy to reduce brain inflammation. It can also slow memory decline and possibly prevent dementia. So we are currently testing these vesicles in late onset models of Alzheimer's disease, which closely mirror the sporadic form of the disease. There are two types of Alzheimer's disease: the familial Alzheimer's disease, which is because of genetics, and then you have the sporadic Alzheimer's disease, which is because of multiple lifestyle factors. So usually sporadic Alzheimer's disease affects 80% of patients. This is particularly important because most people with the Alzheimer's develop it after age 65, and understanding how the treatment works in this population is critical for real-world application. So that is an ongoing study. The second area of emphasis is evaluating how well these CVs work during the middle and advanced stages of Alzheimer's disease. So this would involve testing whether extracellular vesicles can still help brain cells even after the disease has progressed significantly. So if successful, these research efforts will have a tremendous impact on multiple groups of people. First, the treatment could potentially prevent Alzheimer's in high-risk individuals, such as those experiencing age-related mild cognitive impairment or people who carry specific genes that increase their risk of developing Alzheimer's. And second, the therapy could slow down the cognitive and emotional decline in patients who have been newly diagnosed with the disease. And the third one is the most difficult one, that is testing whether this treatment approach could actually reverse some of the cognitive impairments in patients who are already in the more advanced stage of Alzheimer's disease. So this is a summary of one of my major projects in the lab right now.
Well, Ash, I think this work is so exciting and definitely has tremendous potential to have a huge impact as the numbers of people with Alzheimer's, dementia, and cognitive decline just continue to climb out there. And this idea of nasal delivery is really interesting. And it's a drug delivery method that I think a lot of us don't often think about.
I agree. Yeah.
Very cool, Ash. And I know in order to make these exciting discoveries in science, you need to have the right tools and supplies in your lab. And we'd like to take a moment to thank our sponsor, Innovative Research, and to talk about their portfolio of human biological products. Innovative Research is well known for their human whole blood, plasma, and serum, but they also supply other human biologicals and matrices such as saliva, tears, sweat, and urine. Available donations from FDA-approved collection centers range from healthy adults to samples from individuals with specific diseases. Contact them via email at sales@innov-research.com. That's sales@innov-research.com to find out how they can provide human biologicals for your research study today. And going back to our conversation with Ash, I know no research project is without challenges. And Ash, we talked about your work so far in a way that makes it sound like everything has gone smoothly. But every day there are these challenges and struggles that you face. So Ash, I'd love to highlight those as well, because I think it's important for listeners to hear those stories. So do you have an example of a struggle or a failure that you could share with us?
Yeah, there are times I really struggled, but I would like to mention one particular struggle I had in my career. So I got my first R01 grant on my very first try. Some colleagues joked at that time that it was beginner's luck. Nonetheless, I was thrilled and focused on my research, working hard on all the experiments I had proposed and getting the results published during the grant period. So I was focused on what I wrote in the proposal and published those findings. But when it was time to renew the grant, I didn't have enough new data to support my next big ideas for the application. So my renewal application didn't even get discussed. So it was devastating. I was really disappointed. And with the research funds running out, I had to let go of my postdoc and technician. So suddenly my lab was just me. I was the PI, the postdoc, and the technician all at once. That was very difficult. So looking back, I realized that I was a bit naive. So I didn't know then that in addition to finishing what you propose, you also need to do some extra studies to support new ideas for the renewal. So I learned it hard way. So luckily, I still had some funding from the Department of Veterans. So I used that to do some new experiments and gather the preliminary data I needed. And over the next 18 months, I submitted 3 different R01 proposals. It finally paid off. I ended up getting 2 R01 grants at almost the same time. So since then, I've been lucky to keep steady funding from agencies like the NIH, Department of Defense, or Veterans Affairs. But that was a patch in my career where I really struggled, and my lab was just me for almost 2 years.
Yeah, I think that is tough. And how did you, I guess, work through that difficult time? What kept you from quitting and just doing something else altogether?
I'm not a quitter. I mean, that's been my trait. That has helped me. So I just focused on it. I didn't have a plan B. I think that helped. I just wanted to succeed. So I ended up succeeding.
Well, I'm glad this story has a happy ending. And it sounds like 2 R01 grants at the same time is sort of a different sort of struggle.
At the same time. And then I had to hire 3 postdocs simultaneously.
Spin up very quickly.
Yes.
Well, we love to celebrate these successes in science. And I think the success stories are so important because oftentimes so much work and struggle and toil and turmoil goes into them. So do you have a favorite success story, Ash, that you'd like to share with us from your career, whether it was a big win or just a small but really meaningful one?
I totally agree with you. It is very important to celebrate the little wins in science as well as the big wins. And I also tell my research scientists, postdocs, and students to celebrate every success, big or small. So I had some successes, but recently I came to know that one of the original research articles we published in 2023 in Brain Behavior and Immunity on traumatic brain injury, it received the highest number of citations during the 2-year period. And our article has been selected to receive the 2026 Brain Behavior and Immunity Impact Award.
Oh, wow.
Which I will be receiving at the annual meeting of Psychoneuroimmunology Conference next month in New Orleans. This particular paper became very popular and received more than 100 citations in the first 2 years of its publication.
Oh, that is amazing. So how did you celebrate, or have you celebrated yet?
You haven't celebrated this one yet, but we are very excited about it once I receive the award, and we will have a big celebration in the lab with all the authors and co-authors.
Oh, wonderful. And did you anticipate that this paper would have such a big impact when you were writing and publishing it?
We know that it's a very good paper and it had a significant amount of innovative data, but I didn't anticipate that it would be, become so popular. Yeah.
Very cool. Well, congratulations to you and your lab and everybody that worked on this. I
Thank
think
you very
that's so
much.
exciting.
Yeah.
And you do have to make sure you celebrate. Okay, put that on your to-do list, Ash.
Yeah, we will definitely celebrate this one. Yeah.
Well, that is wonderful. And I think these successes can be very motivating and can encourage you to kind of get back to work quickly and just move through that to-do list. And we mentioned earlier that free time can be hard to find as a scientist, but we try to encourage our listeners to take a break, to pick up a book and expand their mind. So do you have a favorite book of yours that you'd like to share with us, or one that you've read recently that you can recommend?
Besides science books and articles, I just don't have much time these days to read anything else, unfortunately.
Mm-hmm.
So back in my Ph.D. and postdoc days, I loved reading the textbook, The Principles of Neuroscience by Eric Kandel.
Mm-hmm.
It's still my all-time favorite neuroscience textbook. Other than that, I keep up with the Science magazine, which I subscribe to. I read it every week. And most of my reading these days is original research articles in my own field. So I don't really have any popular favorite book.
Well, you'll have to ask your lab members, get some book recommendations from them, right?
Yes, I need to do that. Yeah.
Put it on your summer reading list. Well, we'll definitely put Eric Kandel's book on our website if our listeners are looking for a good, solid neuroscience textbook. I've got one on my shelf as well here.
That's great.
Absolutely. And we love to talk about travel that scientists get to do as well. And we touched on it a little bit just in talking about your career path. I mean, your training took you to different parts of India and also to the United States. And I think conferences and working with collaborators and colleagues all over the world is definitely a perk of the job in science. So Ash, do you have a favorite place that your science has taken you that you want to tell us about today?
One of the things I really enjoyed over the years is traveling all over the world for scientific meetings and conferences. Not only do I get to share my research and hear feedback from peers, but I also get to see new places and experience different cultures. So some of my favorite trips have been to Crete, Greece, and Taormina in Sicily, Italy. So I went there for scientific meetings on neural stem cells and the other one for a meeting focused on hippocampus. So in both places, I had amazing conversations with fellow scientists. Some of them are top leaders in the field. I enjoyed beautiful scenery and loved the fantastic Mediterranean food and wine in Crete. and the delicious Italian food and wine in Taormina, Sicily. So these two places I really remember. Of course, I've been to many other places and I like them too.
Definitely. And if our listeners are able to travel to Crete, were you able to do any sightseeing while you're there to recommend some sites?
Oh, it's an island. So basically you can see the entire island in a day or two.
So are you a beach goer? Did you enjoy kind of spending time?
Yes, I enjoyed the beach also. And the food is amazing. Yeah.
Wonderful. And what about Sicily? Were there particular things that stuck out to you?
I have been to only that particular place, the Taormina, and this is a very beautiful place. The food is amazing and the scenery, sightseeing opportunities are multiple. The beaches are great too.
Fantastic. So then how were the conferences there?
The conferences, they don't have a very tight schedule. So usually they have a morning session and an afternoon session, which ends early so that people can go to beaches. And also in between the morning session, afterwards, there's also a long break so that you can spend some time in the vicinity and also go to a beach. So it was pretty low key. But when science is discussed, it's of course quite serious. Yeah.
Definitely. Well, I love that. I love these professional meetings where they build in time for you to get out of the venue for a little bit, do some networking, perhaps spend time with colleagues, enjoying a good lunch, things like that.
Exactly. Yeah. They provided lots of time for networking and as well as sightseeing.
Fantastic. Well, these sound like great opportunities. And I think not only do you get to go to cool places as a scientist, but you get to work with some amazing people. We've talked about some of your lab members, your mentors, and just these people who've had an impact on your career. And I think, unfortunately, there are still stereotypes about how scientists are and what life as a scientist is like. And we try to break some of those stereotypes on our show by just showcasing the human side of science and just the multifaceted, fun, funny nature of the people who are doing the work that we read about. So do you have any stories of quirky traditions or just fun or funny memories that you can share with us that really showcase this human side of science?
So in one of the labs I worked in, there is something called the publication ritual.
Ooh.
When my first paper was accepted in that lab, the PI came to me and told me in a loud voice, Dr. Shetty, we are published together now. So excited. He took me to a bar that evening with other co-authors for a beer drinking session and to discuss the progress I had made on the next paper submission. So it was, I had a bit of a cultural shock then and was not used to that kind of celebration during my Ph.D. student days in India.
Mm-hmm.
Later on, I learned from lab members that was a publication ritual in that lab. So now as a PI myself, I like to celebrate with my co-authors by going out for lunch or dinner or whenever we get an important paper accepted in a top journal. So we do that. Yeah.
I love that. And I think every lab kind of develops its own culture. And I think you're absolutely right. When you travel all over the world, it sort of is this cultural shift fitting into different lab environments. So what was your experience like, I guess, when you made that transition from doing your PhD in India to doing your postdoc first at the University of Montana and then at Duke?
It was a big change for me. It took a while to adjust, but I mingled with the graduate students and other postdocs, and that made it much easier.
Absolutely. And I love to hear that your lab has continued this fun tradition. Are there any other things that your lab does to kind of get outside together and engage in different activities?
Yeah, we do that. Once in a month, we have a happy hour. We go out and have a small party. So I keep that tradition going.
Fantastic. Well, I think this is a fun way to get to know people outside of the lab and very important for building that lab environment.
Exactly. Yeah. So beyond science, we can discuss other topics freely. So that's really nice. And everyone enjoys that kind of interaction.
Absolutely. And I think it gives you some opportunities to sort of take a step back and consider the big questions that you're working toward. And your lab is working towards some very important questions. But as we mentioned earlier, sometimes things like funding can be a limitation, or staff or technology or feasibility. And often these hold you back, Ash, in terms of the questions you're able to answer in the lab. So if we took away the normal barriers and we gave you all of the resources you could dream up, what is the one research question that you would want to answer most?
I think I want to develop an efficient therapy for preventing and curing Alzheimer's disease. So that's my dream. But there are many, many steps there. More funding is required for that. So we need to generate clinical grade extracellular vesicles, get FDA approval, and then go for clinical trials. So it will involve not only funding, but also collaborations with biotech companies and things like that. But that's my dream. I would like to take this to clinic someday.
Oh, fantastic. So do you aspire to get involved in biotech, or you think you're going to stay on the academic side of things?
I'm going to stay on academic side of things, but I would like collaborations because taking any therapy for clinical trials, say, you really need the help of the biotech. It's difficult to do in an academic environment.
Definitely. So I guess what do you see as the biggest barriers right now to making that leap?
The first one is funding. Funding is, I think, the most important issue. Once that is resolved, then the collaborations will happen and then we'll be able to move on from there.
That makes sense. Well, thank you for dreaming big with us and sharing this important project that you are working towards every day. And I think in your lab, there's a lot of work to do, we mentioned. And I think when you get to these difficult periods, it can be helpful to get advice and to have advice from people along the course of your career to help you through all of the different transitions. So for our listeners out there who may be going through some of their own challenges, Ash, is there a piece of advice that you received at some point during your life or your career that you can pay forward and share with listeners today?
Oh, yeah. I received lots of advice. Yeah. So regarding research, at the beginning of my career, when I had anxiety about the outcome of my experiments, my advisor told me, if you know what kind of results you're getting from your experiments, it is not called research. And that was the best advice I got. And after that, I never bothered about the outcome of my experiments. I only focused on the process. The results would come and then you would need to interpret whatever results come, right? That is one of the best advice I received. So now I tell the same thing to new graduate students in my lab exhibiting anxiety about the outcome of their experiments. Another important piece of advice I received is right in the beginning and during my postdoc days also, read a lot within your field. So because ideas come from reading, I really agree with that. And I always try to keep up with the literature as best as I can.
Well, these are both excellent pieces of advice. I'd like to go back to this first one, this idea of not being anxious about how the experiments are going to turn out. I think that is such a good point because the data are going to be what they are, right? Being anxious about it isn't going to change anything. So you may as well just...
Yes. People realize after performing research for a while, initially they come up with the hypothesis and they would think that they would prove the hypothesis every time, but it doesn't happen in reality.
Absolutely. And I love this idea of making sure you're able to read as much as you can. I think particularly in those early stages of your career when you're trying to learn, it's sort of an uphill battle, I think, getting familiar with all the literature and being able to put all those pieces together. So very helpful advice for listeners out there.
Absolutely. I followed that advice.
And look where you ended up. It worked.
Yes.
Well, Ash, thank you for sharing this advice with all of us today. Is there any other last piece of advice you'd like to share or a last note of inspiration that you want to end our conversation on today?
So my advice to young scientists, follow your curiosity. Any problem can become interesting if you look at it very closely. Choose a research topic you truly care about instead of just following the trend or what is popular. So that's my advice.
I think that's wonderful advice. And I think if you pick something that you are inherently passionate about and excited about, like you said, it's not going to feel like you're working.
That's exactly true. Yeah.
Well, wonderful message to end on. Ash, if our listeners want to learn more about you, your work, and some of the things we talked about today, what is the best way for them to get more information?
The best way to reach me is via email. I try my best to reply to emails within a reasonable period of time. And you can also get information about my research at Texas A&M University website and Google Scholar. And of course, you can always do a PubMed research and find the original research articles which we have published recently.
Fantastic. Well, listeners, definitely take some time to check out the Texas A&M University website. Learn more about Ash's research through PubMed, through Google Scholar. And Ash, we appreciate you taking the time to speak with all of us today and to share a piece of your story.
Thank you very much for having me. Greatly appreciate it.
It was wonderful to have you with us. And listeners, always great to have you here with us as well. We hope you'll join us again for our next episode of People Behind the Science.